Hi, Erin.
Hi, Shu. How are you doing?
I'm good. How are
Good
you?
to see you. I'm good. Thanks for coming.
Sure. Love the opportunity here. Should I try my slides?
Yeah, that'd be great.
Can you see it?
Yeah.
Is it in the presenter view or the audience view?
The audience view. Yeah, it looks fine. Looks
Okay.
good.
That's the only thing you need to make sure.
Yeah, thanks.
Awesome. All right, Christina's here.
Oh, great. It's a UCSD day today.
Yeah, that's right.
Hey, Christina.
Hi, Erin. How you doing?
Good. I'm in Shanghai.
Oh, wow. Okay. So it's morning.
Yeah.
So good morning.
Are you there for a meeting or something?
I've been here a couple years.
Wow.
You flying back and forth or?
That's the plan. It's hard now because I mean, when I saw you this summer, I came back for the summer, but then I just flew back. So it's hard to fly back because there's a 10-day quarantine.
Oh, I see. Wow. Are you pretty restricted in traveling?
Yeah, yes, but not. Yes, my kids go to school in Shanghai, so there's a lot of restrictions. They don't want you really traveling around.
Yeah.
Well, I didn't know that's a big news. When did that happen? Like your
last year. Yeah.
Oh, wow. That must be fun. I haven't been back to China for 8 years.
Oh, yeah. You can't come back now, but maybe next year it'll be easier.
You know, we talk about that every year. Still like that.
Yeah. Christina, do you want to test out your slides?
Sure. Am I going to go first or you?
Second, please.
Second. Okay, sure. Share.
Looks good.
Oh,
Wait, that's the different. Yeah, that's the present.
Oh, okay. All right.
Do you know how to swap it?
I'm on my laptop. So.
It was good, and then it switched.
Okay, let me see.
I think if you just do slideshow, and then there's something on the top left after you do that to switch.
Okay, let's see. Usually it's under advanced, but let's see.
if you just hit play, maybe it's gonna go okay. Because it was okay.
Yeah, that's good.
Okay. Yeah, don't switch.
Okay. Okay.
Yeah, that's good.
Great.
Hey.
Hello.
Hi, Xu.
Hi, Zero.
Nice to see you, Christina.
Hello. Hi.
Hi. I was working as a postdoc at UCSD for 6 years.
Oh, I didn't know that.
In biology.
Oh, great.
The Pacific Hall, the ground floor.
Oh.
There's a building near the ocean.
Yeah, my husband works there now.
Oh.
Mm-hmm.
Yeah, we have a lot of colleagues in Pac Hall.
Oh, okay.
My husband is Enfu Hui. He's in biology now.
Great.
I'm coming to UCSD in January for a seminar.
Oh, great.
Oh, nice.
For which department?
I don't know.
Is it neuroscience?
I don't know.
Because I kind of remember seeing your name somewhere.
Some graduate student.
Yeah. It might be neuroscience.
Yeah, sounds about right.
Yeah.
Yeah, I just saw their speakers list. And I think I saw Erin.
Okay.
Are you guys coming to SFN?
No, we can't really do that.
Yeah.
Maybe next year.
Yeah.
But I'm organizing a Cold Spring Harbor meeting in New York at end of November, which I'll go to.
Oh,
Okay. Yeah, it seems like we see you over here every once in a while.
Yeah, my plan is to come back and forth more frequently.
Yeah. Well, the Gordon Conference in Ventura was fantastic.
Oh yeah, it was so much fun. My dog is snoring in the background. I don't know if it's
Can't hear it.
yeah, okay. How's everything going, Zalong?
Yeah, smoothly, not bad.
Good.
So it's gonna be a RS meeting in Shanghai in November?
I have to check. I have to
Oh,
check.
right. You have a New
Beijing.
York meeting. Yeah.
Well, no, I mean, I just have to, I'm going to check at the end of October with
Mhm.
Yunying to see if we can host it there.
Mhm,
I
mhm.
hope to do it November 19th and 20th.
Mhm.
But only if, like right now, I don't think people can travel back and forth from Beijing. And we have a bunch of Beijing speakers.
It should be okay, I think.
I don't know. I tried to book a train ticket to Beijing and it popped up and said you can't travel to Beijing. So maybe
Last week?
no, this week.
This week. Oh, okay. Maybe I still.
Yeah.
But hmm. So what's the topic in Cosmo Hammer meeting? Your group meeting?
Neurodegenerative disease and therapeutics.
Oh, a very interesting therapeutic effort I found associated with neurodegeneration disorder. But for all the developmental, it's so hard, right? It's nobody
Yeah.
really talk about therapy,
Right.
something going on. Mhm.
Right. Yeah, it's been yeah, I'm really optimistic and excited about some new therapeutic efforts.
Mhm.
So we'll hear about them.
Mhm. So the new ARS drug, is that really working?
Is that well, it's approved by FDA. I think
Yeah,
whether it's unclear if it actually has any effect, but
right.
got approved.
Mhm.
It certainly doesn't have a big effect, if any
Okay.
effect.
Yeah.
It's sort of there was like two college students who read some papers and thought, why don't we just mix these two drugs together? And they don't really know the mechanism. They did a clinical trial. They saw a smaller clinical trial. They saw some marginal
Okay.
benefit, but there was a lot of pressure from ALS organizations that
Okay.
influenced the FDA.
Mm-hmm. All right.
So it's something, but I
Need a better one.
need better ones. Yes.
All right, you can start started.
Yeah. Okay.
Mhm.
Okay. Welcome everyone to another week of NeuroZoom. Today's Philadelphia Phillies going to the World Series. NeuroZoom. Sorry, San Diego. And looking forward to great talks from UCSD colleagues today. Before we get started, let me advertise next week's seminars. We have Yi Gu from NIH, and we have Jisong Guan from Shanghai Tech University. And then just an advertisement is that not next week, but the following week, after November 7th, we're gonna, because of daylight saving time ending in the United States, we're gonna, it'll be the same time in the US, but in Asia, it'll be 1 hour later. So in Beijing, Shanghai, we can wake up at 9 a.m., Tokyo, Kyoto, 10 a.m., Taipei, 9 a.m., Singapore, 9 a.m. So just note that. I'll announce that again next week. Okay. Zilong, do you want to introduce our first
Okay.
speaker?
Sure. It's my great pleasure to introduce the Professor Chen. So as I heard, she in high school she was one of the Olympic biology golden medalist, right? Then she went to college in Peking University, where after college she went to US, did a PhD study in Wisconsin medicine, and with Barry Janeski working on the neuromuscular junctions. After PhD in Wisconsin medicine, she went to Gladstone, working with Professor Li Gan, where she started his journey in studying neurodegeneration disorder, particularly focusing on Alzheimer's disease. So after a K9 grant, she started her own lab in UCSD and in school of medicine and the Department of Neuroscience. So today she will talk about the new latest in her lab, motive-based role in AMPK and P300 pathway in tau pathway. Okay, welcome to take it away.
All right, thanks Zilong so much for the very kind introduction. Can everyone see my screen okay?
Sure, I'm gonna put in the slideshow mode. Hey, great.
All right. Well, good morning, good evening, everybody. It's my great pleasure to have the opportunity to present here. So my lab actually started in January 2020 at the peak of pandemics. So I decided today I'm going to just tell you a little bit about what we do in my lab as I first started. And it's kind of building upon my postdoctoral work and beyond. So our current interest and focus. So my lab studies tau. So as we know, tau is one of the two major pathological hallmarks underlying Alzheimer's disease, the other one being amyloid beta. So tau forms those very classic neurofibrillary tangle structure, and those are hyperphosphorylated tau. And such pathology is observed not only in Alzheimer's, but also in a series of neurodegenerative diseases called tauopathies, including CTE and PSP and frontotemporal dementia. So another feature of tauopathy is that this sort of tau pathology starts from a small area of the brain, and it will spread to a larger area as the disease progresses, and that's becoming sort of a focus of the field right now to study how that's regulated. So what makes neuron to go tau crazy? Okay, so tau is normally a microtubule binding protein inside of neurons. And under some insult, for example, genetic mutations on tau or environmental factors, tau can become toxic. And those toxic tau species can go on to form tau aggregates, as we saw in the tangles. And in the meantime, those pre-tangle toxic form of tau and tangles can lead to neuronal impairment and neuronal death. And on the other hand, those toxic tau species can be secreted from those neurons who made them and become those extracellular tau seed. And those seeds, once they are taken up by neighboring neurons, can trigger this sort of a prion-like propagation process. So as we can see, there are two important processes underlying tau pathogenesis. One is the formation of those toxic tau species and accumulation of those tau species, and on the other hand, the tau propagation. However, a lot of those processes are actually not very well understood. For example, what makes tau a toxic protein to begin with? And how do those toxic tau make their way out of the cells are not even clearly understood. So when I first started my lab, I became really interested in understanding the aging-related processes in the whole organism and how they crosstalk and modulate neurodegeneration. So one major focus is to understand the metabolic dysfunction. How does that affect neurodegeneration? So the reason is there is very tight connection between metabolic dysfunction and neurodegeneration. So here is a very striking fact that actually 80% of people with Alzheimer's are known to have insulin resistance or type 2 diabetes. Now, we also know there's a lot of comorbidity of those metabolic diseases phenotype and in the neurodegenerative brain. And on the cellular and tissue level, there's a lot of shared pathological process. Now, we are trying to understand the mechanism underlying such interconnection between metabolism and tau pathogenesis. And on the other hand, are there opportunities for intervention coming from these angles? So my study so far have been focused on tau accumulation and tau propagation. Now, here's an example to show like how tau is really tightly linked to metabolism. So this is a AD brain PET scan. And I hope you can appreciate that in those regions that has higher tau accumulation shown by the tau PET probe are correlated with low metabolism. And such a correlation is not evident in the amyloid beta probe. So this interesting correlation is not shown up only later, it's actually showing up earlier before a major neurodegeneration has happened. So we think instead of being simply a consequence of neuronal death, maybe the metabolic dysregulation are actually contributing or priming the brain tissues towards degeneration in terms of tau. So the story I'm going to tell you about today has to do with this enzyme called P300. And I'm going to tell you some evidence that why we think it represents an important hub of metabolic regulation of tauopathy. So you might have heard about P300 and its close homologue called CBP. So they are best known as transcriptional co-activators, but they also have a lot of known cytosolic functions. So I'll show you some example later. Now, P300 was found as a master regulator in Alzheimer's based on a system biology study. So what my postdoc work and later have shown that P300 can regulate both tau accumulation and tau propagation through either directly acetylation on tau protein or through regulation of autophagy pathway. And at the end, I'm going to show you a little bit of evidence that we think P300 pathway is downstream of a metabolic signaling pathway, AMPK. Okay. So this is sort of when I first joined my postdoc lab, there were several labs together with Li Gan's lab found that tau is undergoing hyperacetylation. So acetylation turned out to be one of a major post-translational modification on tau next to phosphorylation that everybody's talking about. Now, we found that tau undergoes acetylation pretty early on in the disease, and acetylated tau level goes up as AD progresses. And here is acetylated tau antibody staining picture of an AD patient brain, where you can see this classic neurofibrillary tangle structure lighting up, suggesting acetylated tau has something to do with the pathology. So when I joined the lab, I wanted to figure out why does tau become hyperacetylated. So we look into those acetyltransferase, which is the enzyme that catalyzes the acetylation reaction, and I nailed down this enzyme P300. So P300 and CBP, which are homologs, catalyze this acetylation reaction using acetyl-CoA as the substrate, move it onto the lysine residue of the protein. And so what we found is that in AD and PSP, another form of tauopathy, patient brains, there is increased P300 activity found in the CSF, as shown by this surrogate marker of P300 activity, acetylhistone. So here shows the direct evidence how P300 is responsible for tau acetylation. So when I knock out P300 and CBP in primary neurons, I was able to abolish tau acetylation. And interestingly, that comes with a reduced total tau protein level. And this is not due to a reduced tau transcription because the mRNA level was unchanged. So that led us to hypothesize that maybe a hyperacetylated tau has a higher stability and contributes to its higher accumulation. And we actually published that result. I'm not going to go into details. And my colleagues at the same time found that acetylated tau also have toxic role in terms of impaired synaptic plasticity, as well as tau missorting. So next, I want to ask, well, if P300 regulate tau accumulation, how does it regulate tau propagation? So I want to know how does P300 regulate tau secretion or release from neurons. So here it shows in HEX cells, when I overexpress P300 together with tau, not only do I see increased tau accumulation intracellularly, but also there's a massive increase of tau that's getting released into the media, and a lot of which are actually phosphorylated, suggesting that they are the toxic pathological species of tau. So I thought that's interesting because it suggests increased P300 also promote tau secretion. Now the next question is how? What's the mechanism? Well, it's actually not very well understood how is tau released from neurons, but it's been suggested that tau can utilize those intracellular vesicles that are intermediate of autophagic lysosomal pathway and make their way out of the cell. So I decided to test that. So here I treat the primary neurons with lysosomal inhibitor to block autophagy flux. So they're accumulating those intermediate autophagic vesicles as shown by those markers. And I found that not only does intracellular tau level increase, which is unsurprising because autophagy was reduced, but there's also a dramatic increase of tau secretion. So this is relative to the intracellular tau. Okay, so it's percentage of tau that's released to the media. It's increased by 2-fold. So this suggests blocking autophagic flux will increase tau secretion. Now, how's that relevant in vivo? Well, it's well known that in AD brain, there's a lot of accumulation of autophagic vesicles. So these are sort of like a traffic jam that you saw in the dystrophic neurites of the AD brain, where you have a lot of cargoes that's getting stuck in the undegradable autophagic vesicles. And we found similar things in a tau transgenic mouse model, a PS19 transgenic mice, where again, these are the intermediate autophagic vesicle markers are highly increased, suggesting there's reduced autophagic flux in the brain. And so how does P300 might have to do with this? Well, we know that in the tauopathy brain, there's increased P300 activity, and that's also true in a PS19 transgenic mice. So I ran a correlation analysis between P300 activity in the brain and those autophagic markers. And they are tightly correlated, suggesting perhaps increased P300 has something to do with the reduced autophagic flux. And maybe through that, it can promote tau secretion. So this is not a brand new concept. In fact, it's been shown earlier that by several studies that acetylation of many autophagic machinery protein as well as autophagic regulators by P300 can modulate autophagic flux activity. So I decided to test that in neurons. So here I'm using a P300 activator drug, and without going into details, I was able to see a reduction of autophagic flux with P300 activated. And here is another assay using a dual fluorescent labeled LC3 marker, where we can label the mature versus immature autophagic vesicles as a measurement for autophagic flux. And again, we're seeing with P300 activation, there's reduced autophagic flux in neurons. So, of course, I want to know how does that relate to tau secretion. So here I found in a primary neurons that's knocked out of P300 and CBP, there is an increased autophagic flux. So it's the opposite of what we saw earlier. And that leads to a reduction of tau secretion. What about in vivo? So of course, eventually we want to inhibit tau spreading or tau secretion that's coming with it. So here I'm using this model where this is a P300 CBP double flox mice in the background of P301S tau transgenic mice. So what we did is we trigger this tau spreading event by injecting tau fibrils to one side of the hippocampus and watch it spread to the other side, where we inject AAV to express CRE or control locally on this other side and see how much tau spreading is happening. And this is what we saw. With Cre overexpression, we have a reduced tau spreading level. And this again is correlated with a P300 activity. So the higher P300 activity is here, the more tau spreading is. So far I've told you that in tauopathy brains, there is increased P300 activity, and that on the one hand promotes tau acetylation, and on the other hand can inhibit autophagy flux. And together they contribute to both increased tau accumulation as well as other toxic tau species, and also increased tau propagation. So with that, of course, we wanted to see if we can target that hyperactivity of P300 using some drugs. So this was a study that's published earlier when we use this ancient anti-inflammatory drug salsylate, which is found to be a P300 inhibitor. We use them to treat the PS19 mice, and we were able to normalize the increased P300 activity, which leads to reduced acetylation of tau, reduced accumulation of total tau. And moreover, this is the pathology showing that with the treatment, we can reduce the tangle formation, we can prevent the brain atrophy, as well as we can rescue the learning and memory deficit in this mouse model. So this study has led to a clinical trial in PSP patients here. And there were actually a couple very recent publications sort of confirming the importance of targeting acylated tau in one study in the TBI brains. And in this very recent study shows that acylated tau might be underlying the sex difference that we observed in Alzheimer's patients. Okay, so that's sort of the first part. It's more centered around P300 and autophagy pathway. So this is how I came into the metabolic part, because we have all the reason to believe this pathway is underlying a metabolic regulation, because P300 uses acetyl-CoA as its substrate. And as we know, acetyl-CoA is kind of a currency in the metabolic network in the cell. So higher acetyl-CoA means the cells are well fed, and that's how you have increased P300 activity usually. And on the other hand, we also know autophagy is highly responsive to the metabolic status of the cell. So that made me to wonder whether this pathway is under the regulation of AMP kinase, AMPK. And AMPK is known to be an energy sensor and regulator. So it's activated when the cells are starving, so low ATP or low glucose level leads to increased AMPK activation. And we know that AMPK is a strong regulator or promoter of autophagy. So I wanted to ask whether AMPK also regulates P300 and acetylation, and thereby regulating tau pathogenesis. So I took a look at the PS19 mouse brain, and this has been confirmed again in my lab. When we look at the mice as they age, there seems to be a reduction of AMPK activity. And as I showed you earlier, in the old PS19 mice, there's increased P300 activity. So we ran this correlation between AMPK and P300 activity and found they are negatively correlated. So that suggests perhaps AMPK is inhibiting P300, and that might be contributing to why we see a hyperactivity of P300. So to test that, we inhibit AMPK in primary neurons. So here we're using a shRNA to knock down AMPK, and that results in an increased P300 activity. And that is associated with increased tau acetylation and tau accumulation, as we predicted earlier. So what about the flip side? What if we increase the AMPK activity? So here we use a drug ACA to promote AMPK activity in primary neurons, and this time we can inhibit tau acetylation, reduce total tau levels, and that's associated with reduced P300 activity. So again, that suggests AMPK inhibits P300. Now, what about in vivo? So here is an initial test in vivo where we're looking at young PS19. So these mice have not developed a very strong pathology yet. So we are knocking down AMPK by half using the flox over plus alleles and using the neuronal promoter of Cre. So we were able to inhibit AMPK activity, and that leads to increased tau acetylation, total tau accumulation, and again, that's associated with increased P300 activity. And then we did this tau spreading assay again. This time, instead of using P300 flox, we're using AMPK double flox, two alleles of AMPK in the PS19 background. And what we're seeing here is with the Cre-induced AMPK knockdown, there is more tau spreading happened on this side of the brain. So altogether, it seems like AMPK is inhibiting P300, and that could be part of the reason why we're seeing increased P300 activity. Now, we're also exploring the role of this pathway in a Drosophila model. So I was trained as a Drosophila geneticist, so I always dream about going back to my fly models while I'm waiting for my mice experiment. So this is a very simple experiment where we look at the tau flies, where tau is expressing the eye of the fly, resulting in this rough eye phenotype. And we found that when we overexpress AMPK wild type in the neurons, we can rescue this rough eye phenotype, suggesting AMPK is protective in this sense. Now, this is an interesting experiment that I think it actually speaks to how tightly connected of tauopathy to the metabolism in general. So I ran this very simple assay, which is to starve the flies and see how long they live, okay? So I found that there's a very striking reduction in lifespan of the tau flies when they are starved compared to wild type, so they die earlier. And this is very reminiscent of an AMPK loss of function fly that's been reported earlier. So again, that suggests tau flies might have an AMPK dysfunction. Now, strikingly, when I overexpress AMPK in the neurons of those flies, I was able to rescue this reduced lifespan. So those flies are now living almost to the same length of the wild type fly when they are starved. So this really tells us a lot about, there's a lot going on in the whole animal in terms of metabolism and tau. So we are currently doing more study to understand how this pathway is affecting metabolism phenotype in tauopathy animals. So this is kind of what I just told you. So we know P300 seems to be inhibited by AMPK, and we already know AMPK can promote autophagy. So in the tauopathy brains, early on, when there's reduced AMPK activity, that leads to increased P300, reduced autophagy, and together they contribute to tau pathogenesis. Now, I have to say this pathway is way more complicated than I originally thought, because we know metabolic processes are highly dynamic. Okay, so what it means is that we need to know when this regulation is happening, where is it happening, and how. So we're currently looking at if this relationship holds up in aged brain, and is there a difference between acute versus chronic effect? Are there difference in different cell types, neurons versus glias? And on the other hand, are the other mechanisms that's connected to this pathway and adding to this picture. So we're using a series of different models from iPSC neurons to brain slice culture to flies and to mice to try to tease apart these mechanisms. So with that, I will stop, and I'd like to thank my lab members for supporting me through the first couple of years and when I have my own lab, my postdoc mentor, Li Gan, and my collaborators, as well as the funding source. And thank you very much, and we are recruiting, so join us.
Great, thanks, Xu, for a great talk. Now we open for questions. I might start first one. So I'm curious about, I guess people must already ask you before. So what about the other potent inhibitor of MPK, so metformin, so which is a strong metabolic regulator according to your model. So would it be possible to metformin has a natural role in regulating tauopathy in Alzheimer's disorder patients?
Right,
But
right.
they have, yeah,
Metformin is one of the first drugs when I tried in my in vitro
Yeah.
experiment. Yeah, it inhibits
Okay.
acetylation and tau phosphorylation very potently.
Okay.
Yes, I
Wow,
have
okay.
not tried it in vivo yet, but there's a lot of studies showing anti-aging and neuroprotective effect of metformin. So I wouldn't be surprised that we see some result in tauopathy models or patients. Yeah,
It will be opposite, right? So when people already develop the tauopathy, so metformin seems to be something bad.
right. So that comes to what I said, the chronic versus acute effect, also different
Okay.
stage. So to tell you the truth, I believe activating AMPK early on is beneficial, but it's detrimental later on. I don't have time to show the data, but I have some evidence. So you don't want it to activate a whole life. You don't want to boost it when you already have disease.
Okay, great. That's good to know, definitely. Thank you. Yeah. Any more questions?
Hey, Shu, I have a question.
Yes.
Great talk. You showed or you mentioned that new paper that just came out on tau ubiquitination and sex differences with USP11. I thought I'm still digesting that paper, but it seems very interesting. And I'm just wondering, because they propose a competition between the aggregation promoting acetylation, like you demonstrated, and then they're proposing protective roles for ubiquitination. I'm just wondering, in your experiments, either with knockdown or with the inhibitor, when you have less tau, do you see a compensatory increase in ubiquitination? And then also, do you see in your animals, or if you go back and look at the data, more effects in the female mice than the male ones, or vice versa?
Yeah, great question. So actually, ubiquitination is the first mechanism that we explored. So here's actually the data. So originally we thought because it's acylated, it inhibits ubiquitination. Therefore, you have increased tau stability. And this is indeed the case. We're looking at the tau half-life, right? So we saw the acetyl mimic can promote tau half-life to be longer. And also we saw there's competition with ubiquitination. So that's actually one of our major mechanism of the Nature Medicine paper. But it can be very sort of specific to different acetylation site. So that paper was talking about the 274 and 281, and they believe that ubiquitination competition there and USP14 is the one that's responsible for that sex difference. And because of that enzyme is on the X chromosome, right, that explains the sex difference. So I'm very happy to see they follow up on this study. And then, but is that the only mechanism? I'm not sure, to be honest.
Got it. Okay, thank you.
Any other questions?
This is a harder experiment, but have you thought about trying the inhibitor in the tau spreading, in vivo tau spreading model?
Which inhibitor? The
inhibitor? The salicylate?
Right. No, we haven't done that experiment yet. That was more of a chronic feeding and aging model.
Right.
Yeah, it's actually pretty easy to do. The spreading model is shorter. Yeah, but that's a good question, actually. So the one we tried was actually aged animals. So they are older. So if we're talking about this dynamic range and, aged versus young, preventative versus therapeutic, it's worth trying whether salicylate can work for the, sort of younger and acute model of spreading. Yeah.
And there's also a lot of interesting, I think the tau spreading models are very interesting. There's a lot of new work on showing how they can form different strains and how these different strains have different properties. I'm just wondering if acetylation has any role in affecting the quaternary structure or somehow the strains, so you might bias towards one type of strain or another.
Yeah, that's a very good point. Yeah, I haven't looked into how acetylation really alters the structure of those tau fibrils in terms of their species. They are like species, right, of the tau seed. But you're right, they are very specific, and they seem to keep their sort of identity as they propagate. So it's really worth tracking if acetylation changes their identity, and how does that affect the spreading dynamics. Yeah.
Thank you.
Kevin, do you have questions?
Hi, Xu. Nice to meet you. Yeah, nice to see you. So yeah, great talk. So I have a question regarding your last survival curve of the tau flies.
Mm-hmm.
So it's very interesting that tau flies survive much lesser when facing starving, and which can be rescued by overexpressing AMP, activating AMP. So do you think the tau deficient, is that a tau overexpressed fly?
Yes.
Okay, so how does the tau overexpression affects the energy metabolisms in the flies? That is like, so the tau is probably overexpressed across the whole body of the flies, right? Including probably the intestine and the stomach. And yeah, I'm just wondering how tau overexpression may affect the energy supplies.
Right. Yeah, good question. So here in that particular model, we only overexpress in the neurons. So just the neurons. Yes,
Okay.
but
So that
what
would
you said
make it.
was completely valid. No. But what you said was completely valid, because there is a group that reported their cell non-autonomous effect of tau, not tau, sorry, of AMPK. So if you overexpress AMPK in the neurons, it's affecting the pathway in the gut and vice versa. So I think that's super fascinating. So what could be happening here is when you overexpress tau in the neurons, it might regulate AMPK locally, but that can regulate something in the peripheral, for example, the gut, based on that mechanism. Yeah.
Okay, that's very interesting. Okay, great, good talk. Thank you.
Thank you.
Right. Thanks you for great talk.
Thank you.
Amazing. Now, Aaron, do you want to introduce Christina?
Great. Okay, thank you, Xu. And now it's my pleasure to introduce our next speaker, also from UCSD, Dr. Christina Sigurdson. Christina received her undergraduate degree at the University of California, Riverside, and then did a veterinary medicine doctoral degree. So she's a veterinarian at University of California, Davis. And she also did a PhD at Colorado State University with Dr. Ed Hoover. And here she started working on prions and especially chronic wasting disease in deer, which is a prion disease in deer and elk. And she made important discoveries about how prions are transmitted in the wild. She then did a postdoctoral fellowship in Zurich in Switzerland with Dr. Adriano Aguzzi, where she continued to make insights into how prions are transmitted using transgenic mice and also studying ways it may be transmitted in the wild. And then importantly, her and her colleagues developed new fluorescent thiophene derivatives that can be used to image aggregates in vivo in the brain. And these have been powerful tools for studying not just prions, but other protein aggregation diseases. She's started her own laboratory at UCSD, where she continues to have a laboratory and make important contributions, and she's a professor, and we're looking forward to hearing her latest. Go ahead, Christina.
Thank you, Aaron. Let's get it into there. How does that look?
Great.
Okay, great. Thank you, Aaron, for your introduction, and hello, everyone. It's great to be here on NeuroZoom. This has been such a fantastic forum, and I thank you all for participating and joining. So I'm really excited to tell you about a new project that we've been working on. It's not published yet. It's new data, and we're looking here at how prion aggregates are impacting endosomal trafficking pathways. So our lab's been focusing on identifying the drivers of neurodegenerative disease and particularly understanding how these protein aggregates are leading to neurotoxicity. And here, as you also mentioned, you can see that in Alzheimer's, there's just this really tremendous atrophy of the brain caused by the aggregation of amyloid beta, and here you can see it, amyloid beta aggregating in these extracellular plaques, as well as the intracellular aggregation of tau. And a lot of these patients also have aggregation of alpha-synuclein. So prion diseases are actually similar to other neurodegenerative diseases, but they have some advantages in terms of being simpler to study, because in the case of prion diseases, it's often just the one protein that's prion protein that's aggregating. Sometimes there's tau aggregates with certain familiar cases, but it's typically in the sporadic CJD cases, it's only the prion protein that's aggregating. And the animal models represent the disease very well. And in prion disease, the disease course is highly predictable, which is a huge advantage because it means that manipulations are pretty easily interpreted. And I'm going to show you some examples of that. So it's a tool that we can use to understand how protein aggregation can lead to toxicity and disease progression. So I first wanted to show you some recent results that we have from a longitudinal study that we did in mice, where we basically infect the mice with prions just by intracerebral inoculation of prion aggregates into the parietal cortex. And the prions spread throughout the brain. What you can see here in this histology, this is the CA1 region, is that by 40%, we are already seeing ubiquitinated inclusions so early. And you can see that this is a time point before we're seeing any spongiform change in the hippocampus or microgliosis or even astrogliosis. So one of the first changes is ubiquitin aggregation, at least that we can see histologically. Let's see if I can get this to advance. There we go. The prion aggregates are there before 40%, but we're able to see them, and then they really increase exponentially from that time point. So what we did is considering the effect that we were seeing by 40%, we wanted to do a bulk RNA-seq on the hippocampus to see the transcripts that were altered. And what was happening basically at the time where these ubiquitinated proteins were building up. And one of the most significant changes was an increase in immediate early genes, including ARC transcripts, which were up. And we reproduced that just with qRT-PCR. And we also looked at the protein levels of ARC and found that the ARC protein level was increasing at this really early time point. And so while ubiquitinated proteins are increasing, ARC levels are also increasing in the hippocampus. And this was before we saw any detectable inflammatory markers. And this was all done by Jessica Lawrence, who's now at Genentech, in collaboration with Daniel Ojeda Juarez, who's now a postdoc in the lab and did some work on this study as well. So we saw ARC, interestingly, continue to increase as well, all the way to terminal disease. So this early buildup of ubiquitinated proteins suggests that there's a dysfunction in proteostatic pathways. And I really like this schematic on proteostasis from Ulrich Hartl and colleagues. And the focus of my talk today is going to be on this, this part of the proteostatic pathway, which is aggregation and clearance. And here, what you can see are high magnification sections, and just to show you that these prion aggregates can be quite diverse. I think you can see here in a familial case, we see these big plaque-like aggregates. And in this case, we see more diffuse aggregates that's more commonly seen in the sporadic CJD cases. So they can be quite variable. Just to tell you a little bit about the normal prion protein, it's a GPI anchored protein. So it's anchored in the outer leaflet of the plasma membrane, glycosylated. And the aggregated form, PRP scrapie, so this is referred to as PRPC. PRP scrapie interacts with the PRPC form and causes a conversion of PRPC either on the plasma membrane or within the endosomal pathway. And that was work done by several labs, actually decades ago, and a lot of work done on that. showing these two places as important. And then in 2015, this paper came out from Lewis Green's lab at NIH, where they used prion-infected neuroblastoma cells and reported that actually within the endosomal pathway, it's the multivesicular body that's really important for prion conversion. This, they reported, was the site of prion conversion. And this was really interesting because they could block entry of proteins into the multivesicular body and actually drop prion conversion down pretty remarkably. They reduced it by about 70%. So this pathway is pretty interesting because prions have been found in exosomes, and this is kind of at the junction of multivesicular bodies where these interluminal vesicles can be released as exosomes or can fuse with lysosomes for degradation. So, and prions have been found in exosomes, so it really does support that prions are moving through this pathway. And so what we wanted to know with this work was, given that prions are moving through this endosomal pathway, how does the prion protein aggregation actually impact the endocytic pathway? And so the first thing we did, actually Jessica did this work, Jessica Lawrence. She really drove this study. She first just surveyed a lot of the endolysosomal proteins in prion-infected mice. And the most striking finding that she found was that HRS, which is part of the ESCRT complex, was markedly reduced in prion-infected mice with terminal disease. So it was reduced by about 80% for a prion strain that makes these more diffuse aggregates called 22L. But it turns out that this was not strain specific. It basically, all the different prion strains that she looked at, forming either these more diffuse aggregates that are more membrane bound or the plaque-like aggregates, it was the same. Basically, we looked at everything. We saw a big reduction in HRS. And even the few exceptions you can see here, this particular mouse did not have the reduction. Well, it turned out this mouse developed a concurrent disease and was sacrificed much earlier than the other ones. So it was quite consistent. And Jessica also found that its partner protein, STAM1, was also reduced, and HRS levels actually influenced STAM1 levels. So it would be expected that with low HRS, we would also see reduced STAM1. Interestingly, TSG101, which is part of ESCRT1, was slightly elevated, and CHIMP2B, which is part of the ESCRT3 complex, was unaffected. So you might be wondering, what about in humans? And we did look at that, and in the sporadic CJD cases, we also saw a reduction in HRS levels for many of the cases. This was significant, but you can see that there was certain patients that didn't show such a decrease. So this reduction was not due to a decrease in transcripts that we were able to detect, because the transcript levels were unaffected. So what is HRS? HRS is a really interesting protein. It's part of a complex. It's part of ESCRT0, and ESCRT0 bridges endocytosis and multivesicular body biogenesis. These are ancient proteins, highly conserved. They have a 5-domain that the protein uses to bind to early endosomes, and then they have ubiquitin interacting motifs. And so this ESCRT0 complex is really important for sorting ubiquitinated proteins and helping to clear membrane proteins for either fusing with lysosomes and clearing through degradation, or being released as exosomes. And this is occurring in neurons, out in the axons, and in the dendrites. So we were interested to know how HRS depletion, how these low levels of HRS that we see in prion infection would actually impact disease. So what she did was deplete HRS in a cell-specific manner, first reducing in astrocytes, microglia, neurons by crossing the HRS flux mice with different Cree animals, and then inoculated the animals intracerebrally. And what we found was really interesting. So when HRS was depleted from astrocytes, there was no detectable impact in survival, and actually no change in the prion aggregate load. And actually the histology was identical. We didn't see any change in inflammation either. And actually we saw the same thing with depletion of HRS and microglia. Unchanged survival and unchanged prion load by all of our measures. So the difference was actually with depletion of HRS in neurons. So that's where we saw this pronounced impact on survival. So the animals came down with about a 25% acceleration in disease. And although, interestingly, although the mice were developing disease earlier, one possibility would be for some reason they developed a higher level of prions by this early time point, but that was not the case, actually. The load of prions at this time point was actually lower. We wondered whether the kinetics of conversion were altered at all. So we repeated the experiment and then sacrificed the mice at the same time. And at 106 days, we collected all the animals and we actually saw no difference in the PRP scraper levels. So the actual driver of disease at this point was unclear why there was this acceleration. So what we did, we looked at the brain histologically, and what we saw was that even though the mice were coming down earlier, we saw that they did not have an increase in the spongiosis. They actually had a low level of spongiosis. They also had less microgliosis, but interestingly, they had an increase in astrogliosis at this early time point, and that was quite clear, as you can see from these images. So what we have at this time is that we've got, in prion-infected mice, we know there's just to summarize a little bit, there's a big drop in HRS and terminal disease. And when we reduce HRS, we see a decrease in survival time. The mice come down earlier, but by the measures so far that we have, we're not seeing more prions, we're not seeing more spongiform change. The only thing we're really seeing that's a hint is a high level of astrogliosis. So we wondered about the autophagy pathway, because we know from work of Hermann Schatzel and others that that there's a buildup of, at least there's indications that there's a buildup of autophagosomes and a reduction in autophagic flux. As you can see here, we see in the Cre minus mice, so basically the wild type mice, we see, as expected, as it has been reported, a buildup of, or an increase in P62, suggesting that we have a reduction in autophagic flux. We thought maybe depleting HRS would lead to a double hit on autophagy, and we would see a much faster buildup of autophagosomes, but that was not the case. We didn't see evidence of that. So you can see that the level of P62 is actually similar to the controls. And same with LC3 2-to-1 ratios. We did not see an indication that there was a reduction in autophagic flux. We also looked at lysosomes, and here you can see in the wild type mice with the LAMP1 stain, full of lysosomes, but we did not see that in the HRS depleted mice. So we started thinking about just how synaptic degeneration occurs early in prion disease. This is known from the literature. And one of the big functions of HRS is to sort ubiquitinated membrane proteins. So it's possible that the low levels of HRS are actually slowing the turnover of synaptic receptors, including glutamate receptors, which would be expected to have an impact on the synapse. And so we looked at the uninfected animals and the synapses by EM, and here you can see some ultrastructure of the synapse. And we didn't see any impact actually in the uninfected animals. We were a little surprised by that, but we didn't see an impact. And so we then looked at the infected animals, and here's where we saw some really interesting changes. So in the wild type mice infected, what we could see is that over time, we see by terminal disease, a big concave synapses and really big post-synapses, at least in these 2D images, hints of that. thick postsynaptic density, but it wasn't happening until close to terminal disease. Whereas when we looked at the HRS depleted mice, we saw basically the concave synapses were occurring very, very early, even by 84 days post-inoculation. And this is when the mice were starting to show clinical signs. And here I think you can appreciate this big, thick postsynaptic density. And they were terminal by 106 days. So that was interesting. And Jessica quantified these results. And here I think you can appreciate that at terminal disease, the postsynaptic density area, curvature, and length they're not different from each other when they're both at terminal disease, but the difference is at pre-terminal disease, where we're seeing these changes happening early, at such an early time point in the HRS depleted animals. So we started looking at ubiquitinated proteins and found that the ubiquitinated protein level was already high in these animals, even by 106 days, this early time point, and we could visualize these histologically as well. We also looked at 106 days in the prion-infected animals, and actually by 106 days, we could already see that the level of ubiquitinated proteins was higher in the HRS depleted animals. So they were really accumulating a lot of ubiquitinated proteins. So we then wanted to know, well, were they accumulating more ubiquitinated proteins at the synapse? So we collected synaptosomes from prion-infected animals at the same time point. And although there were no changes in the synaptophysin levels, which is suggesting that the presynapse didn't have any changes at this time, we did see higher levels of ubiquitinated proteins in the synaptosomes, suggesting that there was this faster and higher buildup of ubiquitinated proteins right at the synapse. So that got us thinking about AMPA receptors, basically, and what was happening at the synapse. And this is a schematic by my colleague, Gentry Patrick, who's been collaborating with us on these studies. And Gentry's group has reported that mammalian AMPA receptors are bound by ubiquitin, and that is one of the mechanisms that AMPA receptors are actually cleared from the synapse. So we became really interested in what may be happening with AMPA receptors in these animals. And here you can see that basically we looked at the phosphorylated AMPA receptors, especially 831 and 845. 845 in particular is pretty interesting because the GLU-A1 is phosphorylated at 845 when it's at the cell surface. So it would suggest that the AMPA receptors at the cell surface. And we saw that in prion disease, actually the phosphorylated GLU-A1 at 845 was elevated at terminal disease in both the Cre negative and the Cre positive, but that at this position, even by 106 days post-inoculation, the 845 levels were already elevated. So it was suggesting that there was a lot of AMPA receptors at the cell surface, and you can see how this compares with the control, how much higher these AMPA receptors, phosphorylated AMPA receptors, are compared with the control. So we thought that maybe HRS may be having a role in AMPA receptor turnover at the synapse, and it may be a simplistic explanation, and it may be that it's much more complex, but that's something that we're certainly thinking about. But at this point, HRS depletion appears to be at least exacerbating synaptic events that are already occurring in prion disease. So it doesn't seem like that the depletion of HRS is actually, we didn't see any indication of anything different happening. It was more that the depletion caused a full disease acceleration, an early acceleration. So how could this be happening? I wanted to show you this data from neurons that we had cultured in vitro, looking at whether changing HRS levels actually changed prion levels. So we know that prion survival time is very highly sensitive to PRPC expression levels, just the expression of the normal prion protein. And that higher PRPC levels lead to shorter survival. So we really wanted to see whether we were altering PRPC levels. And here we did not see an indication that the total levels were changed. But when we looked at the surface levels of PRPC, we saw a small increase in the surface levels of PRPC, which suggests that PRPC endocytosis may be altered by these low HRS levels. And so this could be at least one possible explanation for the disease acceleration that we're seeing. So that may be a mechanism. So what we're interested in looking at now is how HRS in general, just in wild type animals, could it, just the levels of HRS impact glutamate receptors at the synaptic membrane. So this has not been examined. And so we're working with Gentry Patrick's group to look at this in more detail. And Masa Porhamse in our lab has just really recently completed this series of Western blots. So we did a study where we injected AAV into mice to cause an overexpression of HRS. And here you can see that just overexpressing HRS has led to a pretty big drop in the phosphorylated GLUA1 845 levels, and although it has not affected the total levels. So it suggests that just increasing HRS can potentially decrease AMPA receptors at the surface. Although we're only looking here at phosphorylated, we're setting up to do some in vitro experiments actually looking at GLUA1 on the surface. So that's quite interesting to us. We're also looking at how it affects PRPC levels. And it leads to the question of whether we could restore HRS levels in prion disease, and would that drive PRPC into the endocytic pathway, pulling it off the membrane, and also dampen these AMPA receptors and pull them off the membrane as well. And would that actually increase lifespan and increase the survival time? We've done one preliminary experiment, but the HRS levels were just very modestly increased. So I think we would need to repeat that experiment and do that experiment again. So just in summary, what I've told you about is that we found that this ESCRT0 complex, which is HRS and STAM1, is markedly reduced in prion disease in our experimental models across the board, was not dependent on the strain type, and in humans. And that a consequence of depleting HRS prior to prion inoculation is that we see this acceleration of the disease, and including an accumulation of ubiquitinated proteins that was happening right at the synapse and happening early. These enlarged postsynapses with thick postsynaptic densities, and an early increase in AMPA receptors, phosphorylated AMPA receptors, which is suggestive of dysregulated glutamate homeostasis or altered excitatory inhibitory balance. So as I mentioned, the same findings we see in prion disease, but happening on an accelerated timeline. So we're very interested in this and are investigating further the role of HRS as a potential driver, or trying to understand how this works. And we also would really like to understand the mechanisms leading to the reduction in HRS levels itself. And with that, I will end and thank my group, particularly Daniel and Jessica and Masa. Patricia Aguilar-Cavo also did a lot of in vitro work on these studies, and Jin Wang, Katrine Soldau, and Adela Malik were very much involved. And my colleague Gentry Patrick, who I mentioned, also Viviana Gradinaru and Timothy Miles, Aaron Sullivan, who we're working with on the AAV studies, and Michael Geschwind at UCSF, and my colleagues at Case Western University for working with us on the prion-infected brain samples, and also Nobuyuki Tanaka, who provided us with the HRS flox mice. So with that, I'll end, and thank you so much, and be happy to take questions.
Thanks, Christina. Awesome talk. We're open for questions now. I might start with one. It looked from the histology like there was a it seemed like a dramatic effect on spongiosis in the ESCRT knockout condition, or a delay in that. Do you think that's an effect on PRP, or some direct effect of the endocytic trafficking or the ESCRT complex in those spongiform changes?
Yeah, so we saw a delay in the spongiform changes occurring. But on the other hand, I actually shouldn't say that it was a delay. We saw that when the mice were terminal, which was at an earlier stage it was only at 106 days post-inoculation, that at that disease time point, they had not developed the spongiform change, or very little. So to me, it would indicate that the spongiform change may not be a major disease driver.
Oh, that's a new concept, right?
Yeah, I think it is. And it's just a hypothesis based on that we're seeing so little spongiform change in these animals, yet they were at terminal stages. So yeah, it's as I said, it's just a hypothesis at this point.
Got it. Other questions? I guess you can't look at those because they're terminal. You can't look at them later just to see if, or maybe have you looked at, when you looked at the other ones earlier, the non-ESCRT knockouts, did you see less spongiosis?
Yeah, we didn't do that comparison histologically, but what we did see when we did a time course of prion disease early on, we did note that the spongiosis occurred in the later stages of disease.
I see. Doesn't Aguzzi have some new paper on like endosomes or endocytic trafficking on, like, I guess this is in vitro, but on forming these vacuole structures or in the spongiform changes?
Yeah, that's a really nice paper actually describing how it's basically the mechanism, the pathway that underlies the spongiosis.
So it might be neat to see if those components are delayed in yours, or I just think it's a neat concept that maybe those changes are much later and not driving disease.
Yeah, absolutely. I think it's a really good point. It'd be worth looking at to see whether they are delayed. You know, we could look at some of those components and see whether they're altered by this time point in both the Cre minus and Cre plus mice.
Got it. Cool. Okay, I don't see any other questions coming up. All right. So thanks, Christina, for joining, and thanks so much. Awesome talk. We'll see everyone next week. Phillies will be in the World Series. The world will be great. Good seeing everyone. Bye.
Thank you. That's great. Thanks, Christina.
Astro.